Thank you so much. Everyone can hear me okay? Great, thank you. So I'm actually going to be presenting a bit of work from my PhD thesis. And before I really delve into that, some of you might actually be asking, what is a ctenophore? When we think about jellies or jellyfish, we tend to think about the cnidarians. Most jellies, of course, aren't fish at all. But when we use the term jellyfish, we're usually talking about the gelatinous zooplankton that are going to sting you, that have what we call nematocysts. And the jellyfish, of the classical type, the cnidarians, use those nematocysts for, of course, predation, also as a protective measure. These are a distinctive phyla of animals that have radial symmetry, and they include classes that are going to be your hydrozoans, things like your hydra, also your siphonophores, like the Portuguese man-of-war, by-the-wind sailors, the scyphozoans, which is a nice mouthful. Those are your typical jellyfish, sea nettles, lion's manes, moon jellies. The anthozoans are our corals, our sea anemones, and then a separate group, the cubozoans, for our box jellyfish and so forth. And I'm not going to be talking at all about them because those are actually not ctenophores. When we look at a nematocyst under an electron scanning microscope, it looks something like this. It's literally a harpoon design that has these, almost like rifling features that's going to drill into the flesh of its prey and emit, one of about 38 neurotoxins. And that, of course, can be extremely frightening. We tend to have some fears associated with it. And that's not a ctenophore. Ctenophores, we're going to see, are non-stinging. They don't actually sting at all. They represent their own discrete phyla. There's actually been some recent research done showing that they might be the first of all of our animals. There's been a lot of debate which animals came first. Our sponges, the cnidarians with their radial symmetry, and then the ctenophores. And the ctenophores are so distinct molecularly, we're kind of wondering if maybe they went on their own path and they might have been the first to exist. They're only found in the marine environments. There's no freshwater ctenophores. And what's great about them, at least with studying them and appreciating them, is they won't sting you. They don't have nematocysts. They don't because they're not cnidarians. Again, it's a different phyla. Instead, they have sticky cells called colloblasts. So they're going to have the prey literally adhere to them, almost like fly paper. And when we look at the ctenophores, one of the other distinguishing features is that they have 8 rows of cilia. They're actually the largest animal to use cilia for locomotion. And this makes them actually absolutely beautiful because these 8 rows of cilia actually is what gives the ctenophore its name. It literally means comb bearer. So you look at a row of cilia, looks almost like, a little comb you'd use to brush your hair. But these refract light. So in this picture here, I'm shining a flashlight on one in a tank. You can kind of see a little bit of that prismatic effect. You look at them in the water during the summer, they'll bloom in abundance. The sunlight hits them just right. They make these beautiful rainbows in the water. If you're watching a deep sea special and you see these undulating rainbows of these deep sea jellies. That's not bioluminescence. That's actually just the prismatic effect of the submersible's lighting or some sort of artificial lighting. We can see it right in direct sunlight, but they do also bioluminesce. So they're beautiful in the day, they're beautiful at night. And the neat thing about them, and at least in terms of my research and what I've been able to focus on, is that they do exhibit this boom and bust population dynamics. They will come into such great abundance because they're mostly water. They're about 96% water. So when they're feeding, it doesn't take much energy to produce eggs. They take all that energy, they put a little into their body mass, the rest is going to go into reproduction. And so they get to a point where they're in such high numbers, they literally eat themselves out of house and home, and then they collapse. So it's this nice boom and bust population dynamics, and we can study that. Now, if we look at the ctenophores that are found on Long Island, we actually have 3 dominant species. Occasionally we'll get a 4th and a 5th. Springtime, which in theory is today, I couldn't tell because of the snow falling outside, but the springtime ctenophore is this. It's commonly called the sea gooseberry. You might have heard about it. We call it Chlorobrachia pileus. Very beautiful. It's almost about the size of a grape, and it's got these trailing tentacles. Again, the tentacles are not going to sting you, but they're lined with those colloblasts. Again, literally like fly paper. In the summer months, and this is a little bit of a misleading statement here because this particular species is found throughout the year, but this will peak and bloom in the summer months, we find Mnemiopsis ladii. And this is going to be the subject of my talk today because Mnemiopsis is going to be very abundant during the summer months when things are spawning, things are reproducing. And then occasionally in the fall, we get this guy. I think these are absolutely beautiful, Varroa ovata. These guys actually eat Mnemiopsis here. So occasionally they'll come in through the inlets. We'll get them into Great South Bay. We'll get them in Long Island Sound. And then that will follow, of course, a really severe collapse of Mnemiopsis. But 3 very different beautiful examples of the ctenophores here. And like I said, I'm going to talk specifically about Mnemiopsis. Now, this is a mouthful. You know, I tend to study words that have 2 letters, where one's silent, you know, ctenophore, Mnemiopsis. But just to give you a little inside scoop about this, this actually means unforgettable sight. Mnem for memory and opsis for sight. So there is a little bit of a translation to just how beautiful these are. But they're extremely important gelatinous predators in our environments, particularly within the plankton. But one of the things I won't talk about today that you want to consider is that even things like hard clams, even things like lobsters and your fish, even a bluefin tuna, all begin as a planktonic organism. You know, clams, before they settle down, are actually floating in the water for about 2 weeks. And during that time, of course, they're subject to predation by Mnemiopsis. So they're absolutely incredibly important predators in the environment, particularly because they're mostly water. They can consume about 70% of their body weight in food and prey per day. They can then take that energy and double in size in one day. I used to put this down as a reference, and I've seen it with my own eyes. And they're also self-fertilizing hermaphrodites. They are both male and female, and the eggs and sperm can combine to produce offspring. And they're absolutely, like I said, beautiful. The way they're going to feed is they're moving through the water. This particular species has these lobes. It moves through the water. It takes in that water and any of the plankton that's within it, and then that prey that's inside that water, of course, will get stuck along those colloblasts. So it's a very effective means of predation. Now, the reason I kind of got involved in this is that very few people were studying ctenophores on Long Island. In fact, prior to my master's work, the last person studying them did work in 85 and 86. And then the last person, and that was Great South Bay, the last person for the Peconic was, I believe, 78 and 79. And what I found was, and this agreed with other areas, is that this species is, in fact, increasing in abundance. And not only is it increasing in abundance, but it's also appearing earlier. Like I said, it's a summertime resident. We usually expect those blooms typically in the late summer, September, actually even into the fall, into October. And what I found that in Great South Bay and Peconic Bay, they're an order of 2 to 5 times greater in abundance than they were historically in the 70s and 80s, and they're occurring 2 to 3 months earlier. And that, of course, can have really important consequences for anything that's spawning during that time. If I'm a hard clam and I'm releasing all my gametes and my larvae are going to be in the water, now suddenly this predator is there, that's going to be a problem. And this agrees with other work. We've seen this in Narragansett Bay. We've seen it recently in Chesapeake Bay. And of course, I've shown it not only during my master's work, but repeatedly with my PhD work in Great South Bay and Peconic. So the recent studies have all focused on the predatory role of Mnemiopsis on the zooplankton. It's very well established that this is an important predator. Where you have Mnemiopsis blooms, you have a severe reduction of the zooplankton. But what people weren't looking at was the influence of the larval Mnemiopsis, the babies, on the microplankton. Things like dinoflagellates, things like ciliates, all of those protozoans, all of those single-celled protists, flagellates, and so forth, as a limiting factor to the ultimate recruitment, because their babies aren't feeding on the baby clams and the baby fish and copepods and things like that. They're feeding on smaller things, and very few people were actually considering that. There was actually only a fair number of people up at University of Rhode Island who were looking at it only on a laboratory level. So what I wanted to do is I wanted to take a look at this, because if we consider the life history of Mnemiopsis, it begins with an egg, and within a day that egg is hatched into what we call a sediped or tentaculate larvae. And that larvae is going to feed on the microplanktons, anything between 20 to 200 microns, again, ciliates, dinoflagellates. And then as it's growing from about half a centimeter to about a centimeter or so, it starts developing those lobes. It actually looks like the sea gooseberry. It's a case of that ontogeny recapitulating phylogeny, if you've ever heard that, where you're kind of tracking the evolutionary order in the developmental stage. So it starts dissolving those tentacles. I don't know how well you can see them. They're actually trailing down here. It develops those lobes, and at this point, it starts to feed on both the microplankton and the mesozooplankton. By the size of about 1.5 centimeters, it's feeding exclusively on that zooplankton. And this, of course, is what everybody focuses on, because this is the obvious stage. This is what people are seeing. Now, what I wanted to consider, though, was the impact of Mnemiopsis on the microplankton community when they're in these high abundance of blooms. And I had 2 sort of thoughts about this. I'm wondering, when we get these blooms of exclusively, or not exclusively, but predominantly the larvae of Mnemiopsis, do the microplankton go down in abundance? Are they having an impact like they are with the mesozooplankton? But the other part of it, and this to me was a little bit more important, was what happens when you get the blooms of adult Mnemiopsis? We know, and it's well established both in the laboratory and the field, that mesozooplankton is going to go down. Again, things like copepods, baby fish, baby shrimp, and so on. But would that lead to a trophic cascade whereby the microplankton is released from predation pressure and they might ultimately increase in the water column? If this were to be the case and the microplankton are increasing during Mnemiopsis blooms, wouldn't this be a case of sort of a self-perpetuating feedback system where now you're providing food for the larvae? And again, not intentionally, but you're basically creating a condition. And I wanted to test this because I've always had a theory about it, just because the severity of the zooplankton being reduced was just so extreme under these bloom conditions. So for my PhD work, I actually went out every week from May through October. Had a great tan, in Great South Bay, 2 sites right south of Sayville and north of the Fire Island Inlet, a little northeast. And I was collecting them by plankton net. I was also collecting the zooplankton, and I was collecting the microplankton. I wanted to take a look at all that. Now, as expected, when I looked at my data, I found that Mnemiopsis was severely influencing the zooplankton. So this line right here, this is the zooplankton, and there's normal, oscillations. They go up and down. They'll drop here, and suddenly they're on the rise, mostly because you have a lot of those baby clams, baby shellfish, baby fish, and so forth. Mnemiopsis, which was always present, it's marked by these gray bars here, was always present, but in very small numbers. Once that zooplankton reaches really good numbers, Mnemiopsis peaks. And boy, does it peak, right? So it comes up. These error bars are just showing the differences between toes. And then again, it starts running out of food, right? So it runs out of food, starts dropping. There's a little increase here. It drops. And then you have almost no, again, they're present but in low numbers, but relatively no Mnemiopsis. And look what happens to zooplankton. Yay, right? We're free. The neat thing with this is 2008. It ended up being, at the start, a 97.9% decline in zooplankton. You would run a tow and you'd expect to get basically what I call snot, in your net, and you get nothing. It was just nothing other than Ctenophores. And the neat thing about the 2008 data is that you get a loss of Mnemiopsis with the recovery of zooplankton because Burrowie actually came in. Burrowie was that carnivorous ctenophore I had shown you that occasionally comes in through the inlet. So as soon as Burrowie came in, it ate up all the Mnemiopsis. The zooplankton were very, very thankful to that. Now, 2009 data, we didn't get Burrowie, but we did have similar trends in that we had a 99.7% decline in the zooplankton. Again, zooplankton's going up and down here. Mnemiopsis was present later, at least as far as I could detect by my nets, present later in the spring, and then it just peaks, right? You get some good amounts of zooplankton, it peaks, and then it drops. And then you had a lingering here. You actually had it extending well into the fall, which I always joke was because I started teaching full-time at Suffolk, and it was just Murphy's Law. I thought I'd be done sampling. Nope, so I had to go out. I would literally leave my class in, my boots and attire ready for the boat. So we knew this, right? This is what we had expected. Now, the neat thing, though, was that during both sampling years, those seasonal population blooms of the adults coincided with, as I expected, these substantial increases in the microplankton in Great South Bay. And what was great about that is that we saw these correlations between high adult abundance of the Ctenophores, low mesozooplankton with high microplankton abundances. And those preceded the increase in the larvae. So we get the increase in the adults, we get the increase in the microplankton, and then the larvae subsequently increase. And it looks something like this. So here is the, just identifying the adult Mnemiopsis here. You get that nice peak here. And then if I take a look at the copepods, a type of the zooplankton, they drop, right? They don't do well at all in the presence of Mnemiopsis. They're a dominant food source for them. And then here's ciliates. So these are our naked ciliates, things excluding tintinnids, if you're familiar at all with ciliates. Dominant microplankton, our dinoflagellates. You get almost 1,000% increase in the ciliates. You get almost a 5,000% increase in the dinoflagellates. You get this really nice demonstration of a trophic cascade, and of course, the increase in the larvae of the Ctenophores follow. And this happened in both years. I'm just showing 2009 for now. I'm actually going to come back to 2008. But this is exactly what I was hoping, because this suggested significant top-down control of microplankton by the larvae, and it had only been demonstrated in the lab at that point, and now we're realizing that, hey, as a whole, this ctenophore is having a huge impact on the entire planktonic community. But the neat thing about it was that, if we look at this, the larvae, of course, are taking down about 45 to 56% decline in dinoflagellates in 2008 and 2009, 83% and 97% decline in those ciliates. Ciliates are a akin to like a Big Mac, where a dinoflagellate's more like a hash brown. You know, the ciliates are very, very meaty, and they're going to be a good source for the ctenophores, especially those larvae. And so what this is showing is that the Mnemiopsis ctenophore is involved in this direct feedback system, just like I had hypothesized, where you have such great abundances, such great densities of the adults, that you're reducing the mesozooplankton, things like copepods, and that is subsequently feeding down the food web to increase the microplankton. Now, when you increase the microplankton, you're actually benefiting the larvae of those ctenophores, because that's what they are feeding on. And that, of course, is going to result in a decrease in microplankton, and it also resulted, I didn't show the data for lack of time, but it also resulted in a further trophic cascade to increase nanoplankton. We actually had an increase in microflagellates associated with the decrease in microplankton. So you had a trophic cascade here, you had a trophic cascade here, and they were feeding into one another. Again, the ctenophores aren't purposefully doing it, you know, it'd be neat if they were, but it's just a consequence. And it's starting to help explain why we get these blooms. Why do suddenly we have a few, and then within days we have so many? You're literally swimming through, you know, a sea of ctenophores, especially in Great South Bay. So this brought me to an additional chapter in my PhD work, but it asked the next question is, what role then do the microplankton play in regulating the recruitment of ctenophores in Great South Bay, right? If they're such an important factor to these population blooms, maybe we should start examining the role of microplankton. And along those same lines, what other factors are influencing the intra and interannual differences in the fecundity, meaning the egg production of adult Mnemiopsis? In other words, why do I have such good egg production in June and not July? Why do I have such good egg production in one year and not another? Why do I have a bloom in one year and not another? So all of these are related. And so in addition to those tows that I was taking, I also individually collected ctenophores for gut content analysis. I didn't want them run through the net. I didn't want to increase any prey unnaturally. I would just dip net for them. Again, I had a great tan. I'd get the ctenophores for gut content, and I'd also bring them back into the laboratory same day, put them in an incubator at the ambient temperature, and overnight see how many eggs that they would produce. And possibly sometimes when the water's colder, wait 48 hours and document all of this. Now, when I looked at this, I found that the number of prey items found in my ctenophores were nearly 3 times greater in 2008 than in 2009. And subsequently, which makes sense, the ctenophores in 2008 produced nearly twice as many eggs than they did in 2009. So looking at the gut, you know, in a number of hundreds of individuals, looking at the egg production in hundreds of individuals in both years, I found that there's this nice correlation. The more you eat, the more eggs you're going to produce. And this was not a novel, you know, finding. This made sense. This agreed with laboratory studies. And here is, you know, sort of the reminder here, because there's going to be a quiz at the end, right? So as a summary, because things are going to get a little dizzying, as a summary, 2008, you had more prey, you produced more eggs. Compared to 2009, you had fewer prey, you produced fewer eggs. Okay, so 2008, we had more eggs, we had more gut content. Egg production and gut content both correlated positively and statistically significant with the size of the ctenophore and the zooplankton abundance. This makes sense, right? If I am a larger ctenophore, I produce more eggs, I eat more. If I am a smaller ctenophore, I produce less eggs, I produce, you know, I eat less. If there's more zooplankton in the water, I'm gonna eat more, I'm gonna have more eggs, right? And so this is what was demonstrated already in laboratory experiments, and it agreed with my field data. This is wonderful, right? The problem is that when I compared 2008 to 2009, neither ctenophore size nor zooplankton abundance differed between those 2 years. So I can't explain why I had more eggs and more food in the guts in 2008 than in 2009. I said there must have been more zooplankton in 2008. Nope. Ctenophores must have been bigger in 2008 than 2009. Nope. So when I'm describing this, and I have here as, you know, discussion, it's really not so much a discussion. It's really more a case of a Mystery Science Theater 3000, where we're trying to sort of mock the discussion that's going on here, because this is where you kind of take this with a grain of salt. I'll try to make sense out of it, I promise. But what I'm trying to get at here is I'm trying to figure out what was different between 2008 and 2009 that led to, again, 2008 having more prey items, more eggs than 2009, where you had fewer prey items, you had fewer eggs. What was different? And so I started looking into the feeding mechanisms of this ctenophore, and that's where I need to explain a little bit here. When I looked at the composition of the gut content, I found that there was a very significant dependence on this particular copepod called Acartia tonsa, very dominant in Great South Bay, as well as Long Island Sound in the summer months. You get a different species, Acartia hudsonica, in the winter, but in the summer months, this is your dominant copepod. Just looks just like plankton and SpongeBob with the one eye. Very, very dominant in my Ctenophores. Not a surprise, right, but I wanted to verify this. Now, when I looked at this between the 2 years, I found all life stages of the copepod. We have nauplii, we have capepodites, we have male and female, and collectively made up about 50% of the total gut content. So 50% Acartia, 50% other things. Now, when I compared the 2 years, I found that, as expected, I had more of the copepod in 2008 in my guts, on average, per individual, than I did in 2009, significantly more. But when I looked at the percentage, I had, again, more in 2008 than in 2009. I wasn't expecting that percentage to change, right? It made sense that, yeah, I knew there was more gut in 2008 than in 2009, but why is my percentage different? And this is where I would be at like breakfast in the morning, and I'd be talking to my fiance. I'm like, I don't get it. I just can't figure it out. You know, it's totally occupying my brain. Because remember, 2008, more prey, more eggs. 2009, fewer prey, fewer eggs. And I can't compensate, or I can't, resolve the difference by the zooplankton density being different, because it wasn't. It was the same in both years, and my Ctenophores were the same size in both years. So what is going on here? Well, this is where, maybe put a little vodka in your soda at lunch. This will help you out. When I really took a look at this and I said, what was different between 2008 and 2009, I almost forgot about this, the brown tide. There was a very significant brown tide bloom in 2008. And I wasn't studying brown tide. Brown tide is actually a nanoplankton. So even, with my microplankton studies, or it's a picoplankton technically, I really wasn't looking at it that much. But we had a tremendous bloom in 2008 of almost 2 million cells per mil at its peak. And so when I thought about this and I said, okay, well, what does that mean? It obviously didn't affect the mesozooplankton. The numbers were the same, right? I saw the same results with the microplankton and the trophic cascade. What is going on? So I started analyzing my data, and I found that the number of prey in my Ctenophores during the high abundances, what we consider the bloom abundances of the brown tide, Oreococcus, correlated positively with the abundance or the density of brown tide. So where we had high abundances of brown tide, I had high numbers of prey in my Ctenophores. Something very interesting there. Brown tide seemed to be benefiting the Ctenophores in their feeding. They were feeding better. And I've always wondered this, because when I enter a brown tide, or I've seen red tides and rust tides, I find a lot of my Ctenophores, always. And it's almost like, am I just identifying them better because the water's murkier and they're so clear compared to it, that contrast, or is there something going on here? And it turns out there is something going on here. So how does this work? Well, remember, with Mnemiopsis, it's a lobate ctenophore. It moves by those cilia rows, right? And it starts in training in water. So as it's moving through the water, it's going to take in prey. If you are, I'm sorry, let me let that roll out. If you are a small copepod, like Oithona here, Oithona is a type of copepod, you are literally going to get sucked into that field, right? And you're going to get gobbled up by this ctenophore. Now, if you are a larger copepod, like Acartia, those ciliary rows, that movement through the water is actually not going to pull in these copepods into the ctenophore. Instead, the copepods are not going to be as affected by that stream of water. They're going to be going about doing their own business. They're going to be swimming along, and they encounter Mnemiopsis by literally swimming into it. They're not getting pulled into it. About 82% of the encounters between the Acartia copepod and Mnemiopsis ctenophore were the result of Acartia literally just swimming right into the ctenophore. Oops, now I'm dead, right? And these were studies done in the lab under high precision photography. And so there was a big difference between Acartia and other copepods here. So I looked into this and I said, okay, well, now I know how Acartia is ending up in my ctenophores. Maybe this will help me. Could the differences in the swimming and feeding behaviors of Acartia between those 2 years explain why I had so many more, including the percentage, right, of the copepod in my Ctenophores in 2008 than in 2009? Well, here's the neat thing. This little tiny copepod, you never thought plankton would be so complex. This little tiny copepod is known to switch swimming and feeding behavior based upon the prey availability. Here's how it works. When you have an abundance of ciliates, those are very, very meaty. They're also swimming, they're ciliated. The copepod uses an ambush strategy. Why waste energy? A ciliate's gonna swim right into me, and it's very, very meaty, right? I'm just gonna sit and wait, then I'm gonna grab it as it comes to me. However, when diatoms and phytoflagellates, which are a little smaller, they're not as mobile, if mobile at all in some cases, they're gonna switch to suspension feeding. That copepod's gonna be using its feeding appendages. It's gonna be using a lot of energy to draw as much phytoplankton in as possible. Okay, now this was demonstrated back in the 90s. So I considered this. In Great South Bay, I looked at my microplankton abundance data, and I found that the flagellate abundance was significantly greater in 2008 than in 2009. So this is starting to make a picture. When I look at the difference between the flagellate abundance and the brown tide abundance, I found that, hey, as brown tide density goes up, microflagellate abundance goes up. And that is helping to explain why there were more flagellates in 08 than 09. Brown tide is changing the entire plankton community, at least on the nanoplankton and certainly the picoplankton level. So now when I take a look at this, here's my data, right? I compared all of my microplankton, diatoms, dinoflagellates, flagellates. The only one that had a result, and a positive result, was the flagellates right here. Ciliates, this is very interesting. Ciliates also had a negative relationship, but they were not statistically significant. So unfortunately, I couldn't use that. But what I could use was another study by my PhD advisor who had found during one of the brown tides in the 90s, there was a relationship between ciliate population growth. It was negative. Where you had brown tide, you had a reduction of ciliates. When I started explaining all this to me, she goes, I've seen that before. Ciliates don't do well with brown tide. Flagellates do. So now let's think about this. Here's 2008, we have our brown tide. Here's Acacia, we've got here's your brown tide, here's your flagellates. These are my lame attempt at ciliates, right? And you've got the ctenophore that's going to be moving through the water, right? You've got more flagellates in this case during the brown tide. That is causing the copepods to switch to suspension feeding. Because they're suspension feeding, they're not sitting and waiting, it's not ambush, they are increasing their encounters with the ctenophore. And that results in an increased feeding success and egg production of the ctenophore. So 2008, just from altering the nanoplankton and the microplankton communities, you're changing the behavior of one of the dominant prey items of the ctenophore. If we compare this to 2009, where there wasn't a bloom, brown tide was present, but not nearly, Oreococcus was present, not technically a tide then, right, not a bloom. You had more ciliates. More ciliates, these copepods, they're just gonna be, sinking through the water column, hoping to encounter ciliates. Why waste the energy? Something's gonna swim into me. You ended up with a reduced feeding success and fecundity of Mnemiopsis. Got that? So my question was, now I'm thinking, all right, now it's sexy, right? Now I've got, jellyfish are taking over the world, brown tides are occurring more and more, right? Do brown tides promote ctenophore blooms in Great South Bay, right? We saw that my ctenophores were 3 times greater in terms of gut content in 2008 when I had the bloom. They were producing twice as many eggs in 2008, right? So do brown tides cause ctenophore blooms? Well, no. My abundance of ctenophores were 5 times greater in 2009, right? Say, what, right? What is going on here? So now I'm back to breakfast, beating my head with, my cereal spoon. So here's the thing. Remember what I was talking about in that first chapter. Larval Mnemiopsis depend upon particular microplanktonic prey. So the abundance and the composition of that microplankton might regulate the recruitment of those larvae into those lobate adults. They might regulate subsequently the timing and the magnitude of those ctenophore blooms into those mesozooplankton feeding adults. What we had in 2008 is we had those ciliates and those dinoflagellates, which are excellent food for the larvae, occurring when we had the larvae, right? So here's my increase in dinoflagellates and ciliates. I have my larvae right here. That's wonderful, right? But here's the problem. The adults were peaking in egg production weeks before we had this right here, and in particularly 1 week before. There was a mismatch. So while my adults are doing great, 2008, that brown tide was wonderful for the adults. They're eating more. They're producing tons and tons of eggs. It's essentially like you're producing all these babies, but your babies are starving, right? So we weren't getting a big bloom, and not at all in 2008 compared to 2009. If I compare this now with 2009 data, again, here's my peak in ciliates and dinoflagellates. That's the same thing, coincident with the adults, right? Here is my follow-up of the larvae, right? So they're feeding on the ciliates, they're feeding on the dinoflagellates. That's great. What are the adults doing? Where's the egg production? Turns out it's a match. So where my adults were producing the most of their eggs corresponded with the timing of the plankton where the larvae had sufficient food. And that was the reason why we had 5 times greater abundance of the ctenophores in 2009 than in 2008. Dizzying, right? So the presence of the brown tide in 2008 may have helped Mnemiopsis reproductive success because it enhanced the feeding. It enhanced the feeding of the ctenophore because it altered the feeding of one of its prey items. Now, the mismatch, however, between the microplanktonic prey for the larvae and the egg production by the adults ultimately resulted in the reduced recruitment. And that magnitude of the ctenophore blooms might depend more on the availability of microplankton, not mesozooplankton, but the microplankton as sort of that stepping stone. I need to feed my babies first, right? I'm not going to get a big population no matter how many people are producing, well, not eggs, but producing babies, right? If those little hatching larvae don't have a sufficient food source, they're not going to then establish themselves and metamorph into those low bait adults that are going to subsequently feed on the mesozooplankton. So if you could follow any of that, right? I love this. I miss Gary Larson, the far side. Only they know the difference, right? So with that, I will gladly take any questions that you might have about this dizzying research that I had hopefully tried to explain to you. Thank you. How might the time? You have a few minutes? Okay. Yes. Yeah.
Did you be able to tie that to the climate change at all?
No, what happens with Mnemiopsis is that they do tend to do better if we have a warmer winter because they are what we call holoplanktonic. They don't settle out like a normal jellyfish would. And so if you have a warmer winter, because they're present in the water year-round, you tend to have more surviving in that warmer winter. And there has been a little trend in Great South Bay, but not terribly significant, of warming winters, obviously not this one and last year. And so there is an aspect or an element of the climate change. The climate change also has to do with the warmer water changing their prey. So if you have the mesozooplankton, the copepods, they're increasing, they're doing better, they're feeding better because now the microplankton are doing better, then your ctenophores are going to increase. So I didn't look at it with climate change. One of the things I wanted to look at was in 2008, my abundance of the ctenophore was almost identical to the previous studies in the 80s. When I looked at these increases and I say, wow, in 2008, it turns out they're not increasing like they were in 05 and 06. I compared it to the 80s data. That was also during a brown tide. So I said, well, that's interesting, right? They're numerically almost the same, and I wondered, if that person who had examined that looked at egg production, would she have found the same thing? But it is kind of neat. You kind of think the worst, brown tide, oh my gosh, Armageddon, the apocalypse, but in terms of ctenophores, their numbers actually go down during it, but not, I didn't, the climate change couldn't explain it in my case. Yeah, thanks. Yes?
Yeah, I was just wondering, what's the main predator of this organism?
Yeah, so the dominant, sort of the worst predator is Varroa. So if there is Varroa in the ecosystem, Varroa will gobble them up, but also the lion's mane. Lion's manes absolutely love it's a gelatinous, it's almost like a dead-end food web. It's all gelatinous. Jellies will also feed on it. The neat thing about the predators is certainly sea turtles will feed on jellyfish as well as the ctenophores. Sea turtles are, of course, down. Their numbers are down. Butterfish is also a specialized predator on jellyfish, including ctenophores. Their numbers are also down. There have been some studies looking at dogfish. Depending upon your research, some people are finding that the dogfish, the sharks, are increasing. Other ones are finding they're down. They also feed on the jellies. So that's one of the problems, is that the predators, the natural predators of these jellies, are actually on the decrease, on the decline, and have been for decades and decades. And that's one of the arguments. There's actually a number of arguments as to why, as a whole, these jellies are increasing. Yeah, it's a good question.
Other questions? Go to the microphone behind you.
I can speak up.
We were trying to record this.
Oh, I see. It's a really important question.
Yeah.
Better be.
I hope. I'm just curious, what else feeds on phytoplankton?
Oh gosh, well,
the
your
other
clams,
planktons.
Obviously all your suspension feeders are going to be taking out the phytoplankton, so the hard clams, the scallops, the soft clams, all of your, copepods, the meroplankton, when you're a larval clam, a larval crab, a larval lobster, whatever that might be, you're going to be taking out the phytoplankton as well. Menhaden and even bunker, they're going to be filtering through. They're getting phytoplankton out. They're going to be taking that just as much as the zooplankton. Yeah.
All right, thank you so much.
Sure, thanks so much.